one parent. Now as we have seen that in the case of asexual reproduction, there is no involvement of gametes, and they will just produce the new individual by the single parent. So there are a lot of types of asexual reproduction is here. First one is binary fission, second one is budding, third one is sporulation is written, or you can say the spore formation. The fourth one is vegetative propagation. Vegetative propagation is a long topic, so just I will conduct it later, but for this, that means in this video I will just complete these things: binary fissions, budding, spore formation, this one regeneration, and the tissue culture. Tissue culture also just I will make another video, but in this video I'm going to conduct the other one. So let's start with the binary fission. So here the binary fission. Now if I would like to tell you about the fission part. What is fission? Fission means to split. So that means in this method what we are doing, a single parent is dividing into two parts. That means that means they are losing their own identity, and they are just making into two part, just like in the, as we all know that amoeba is the unicellular organism. It is just made up of only one single cell. So here we can see this is the only one amoeba. Now in this case they are splitting their cytoplasm, sorry, not cytoplasm, first they are elongating their nucleus. That means they are stretching their nucleus, and they will divide into two. So in the third image we can see that the nucleus is being divided. It is also divided into one part, it is divided into two part, and in the last figure we can see that the full cytoplasm is divided into two cells. Now from here we can see that here it was just only one, but here it is two. That means the reproducer loses his identity, and they made the two daughter cells. So this is called the fission, and here this is the example of binary fission. So I have just shown you the example of amoeba, but if you see the example of paramecium, or that means any other that means organism, over there also you can see that just they will first divide the nucleus, and they will divide the cytoplasm. That means over there also they will just lose their identity. The second one we are having with the fragmentation. This is the example of fragmentation. Now in the fragmentation there is a very good example is there. We don't have a lot of example, but in the fragmentation we are having a simple example, that is the example of Spirogyra. Now Spirogyra is just a long thread like filament which break into two or more daughters. So here I can show you that each of the fragments, that means if this is the full, if this one is the full body of Spirogyra, that means Spirogyra we can see in the ponds. Okay, so they are having the greenish color. So these fragments, that means they will divide these fragments into two part or more than two part. So and that single fragment will generate into a new Spirogyra. So this is called the fragmentation, the splitting of their body, and they just make the daughter fragment. That means the new organism is just growing from their own body. After this one we are having the regeneration. Now what is this regeneration? Let's see. Now in the regeneration part, what is this one? That means the process of regrowing. That means in the regeneration we will see that from a single part that means from the singles, that part, the if they are having the ability to grow, that means to regenerate, so they will regenerate. If over that part, if just like if our leg is being cut, so we don't have the ability of regeneration, so we can't regenerate. But if we see the example of um, what we say, that means lizard. If we take the example of lizard, if lizard is losing its tail, so what is happening? After some time we can see that their tails is coming out. But if you have seen that if their head is being cut, so is the head regenerating? No, because the tail is having the power of regeneration, because over that the cut part they are having the regenerating cell, but in the head part they are not having the regenerating cell. So here the doubt is clear. That means if that part is having the ability of regeneration, the regeneration will occur. If they are not having the regeneration, they will not regenerate. Just like in this case in planaria, if their body is being divided into two, they can regenerate into two. Same thing we can see with the hydra. When the hydra is just cut into two part, they can regenerate into two. But if we cut the single, so can they regenerate? They can't regenerate. The best example is the starfish. So in the starfish you can see, if you cut a single part, this one, so they are having the capability of regeneration in their whole body. So they can, that means with the that means with any cut part, they can regenerate the full organism, full body. So this is the examples of regeneration. The next one we are having the budding. Here I have taken the example of yeast, a mature yeast cell. So here this is the mature yeast cell. Now if there is a, that means if there is a extra growth in the body of the yeast cell, that is called the budding. So if that extra part, if it is cut down, and if it is having the ability to grow and to just give rise to a new daughter cell, so it will be called as budding. That means it will be detached from their body, and then it will grow into a new daughter cell. The other one we are having the spore formation, or as you have seen, it is written the sporangium. So this is the example of a bread. If you just keep a bread, that means in the outside, that means if you take out with that means from their polythene, from their packet, and if you leave it just for 2 to 3 days, after 2 to 3 days you will see a black color molds are being just start growing in your bread. So what are those things? As we all know that they are the rhizopus, they are the fungus that they are growing in the bread. So if you take a closer view, if you put that that means if you take, that means the rhizopus is just growing on that one. So if you see the rhizopus, they just divide themselves with the help of the spore formation. That means here you can see when the rhizopus grown over here, so in this one when they developed itself, so here they are just forming the spore, and through the spore it will be, that means distributed in the whole bread. So up to this one only, today we are going just we will read. After this one we are going to start with the vegetative propagation and the tissue culture. So have a nice day.